The detection of cell surface interactions using currently available biochemical approaches still poses technical challenges. This protocol provides a genetic alternative using a genome-scale cell screening approach to identify the receptor-ligand interactions at the cell surface.
Unlike most other existing methods, this method makes no assumptions regarding the biology of the receptors and provides opportunities to identify the interactions mediated by a wide range of cell surface molecules.
Cell surface molecules are directly accessible to drugs such as monoclonal antibodies. Therefore, identification of interactions mediated by these molecules can have important therapeutic implications.
This method is generally applicable to researchers interested in exploring the cellular signaling and recognition processes in a wide range of biological contexts, including neural and immunological interactions, as well as host pathogen interactions.
The protocol requires a high-throughput cell sorting machine and next-generation sequencing machines. Ensure that these facilities are available before starting the protocol.
Begin by making 8 serial dilutions of the biotinylated protein samples in a 96-well plate, ensuring that the final volume of each dilution is at least 200 microliters and that a tag-only control is included. Make a duplicate plate of the samples by removing 100 microliters from each well and transferring it into a new 96-well plate. Include a tag-only protein control that will be used as a control probe in all binding assays. Add 100 microliters of 0.1 microgram per milliliter strep-dividin PE to the wells in one of the plates, and 100 microliters of dilution buffer to the wells of the other plate. Incubate the plate for 20 minutes at room temperature. Meanwhile, block the wells of a strep-dividin-coated plate with the dilution buffer for 15